Can everyone hear me online? I'll respond. I assume that you guys, yes. Okay, perfect. All right, let's get it started. So I have 45 slides. Given my speaking speed, maybe I will finish in 45 minutes, maybe a little bit more. So today, well, first, thanks for coming. It's the prime summertime and appreciated your attendance. Today I will talk about the peripheral measures in fMRI. I will start with my own view, asking questions, and slowly get how the current standards in the field and what is next and what we do in the lab differently. To start with, I mean, the common question is maybe not the right one. What is happening inside the brain? It just drives many neuroscientific studies. We just focus only on the brain and the parts of the brain. Even visual system or like more confined spaces or like an attention or perception. But the question could be, what is happening in our body? It's just maybe the question is not we're ready yet. Or we just like to isolate the systems because the way we perceive is we divide things into chunks and we try to understand small pieces. So maybe the right question is what's happening in the brain-body system, how brain is interacting with the body. It is a question, maybe we are not technically ready. And maybe the question is, why don't we scan the whole body? I mean, why are we just scanning the brain? If we scan the whole body, we will know what's happening in the peripheries, in the lungs, heart, everything. That will be fantastic to have snapshots of the whole body every second or so, so that we can study brain function together with the body function. Well, things are not easy because of imaging limitations, because the way the MRI works, the way the imaging works, it is confined spaces. The imaging of MRI is optimized in certain body parts. Body tissue has different properties. MR sequence needs to be optimized for certain body parts. So it is a delicate system. When you go in the MR console, you see that, okay, where you want to scan brain. So there are like specific settings for the brain. Even if you prepare your patient like this and you get ready for snapshot of a body, that will take a very long time. It will be a sequential changes of the settings of the scanner. But yeah, I mean, there are limitations to do that. And also, body is different too. That might be the reason why we just focused on the brain. I mean, brain is basically fluid and fat. If you go down, there are like lungs, which is air, and inside, in the middle of the lungs, there's like a fast-moving object, heart, this pumping heart. That changes the things crazily with the tuning of the system. If you go down, all the internal organs moving, there's a peristaltic motion. There's like different contents of it, different contrast, and so on and so forth. So it's just this question we are not ready yet. And just want to talk about a little bit scanner limitations. I mean, because the title of this talk is why we using peripherals. Scanner is a limited thing. You need to decide if you want to go for high resolution, confined spaces, then you need a long scan time and less coverage. Or if you want a whole brain and you want to be fast, then your resolution will be very low. I mean, the common people do whole brain with a decent resolution with the advance of neo-gradient systems and so on. But a lot of questions is like more like high resolution in V1, for example. And that's a huge exclusion right there, assumption of V1 works alone. Actually, there's a lot of external areas in the brain that helps V1 to work, right? So it depends on the question and research thing too. So I'm just trying to say exclusion starts right in the brain already. You're not yet ready that in the body yet. And yesterday preparing this talk, I was looking at the atlases, which is commonly used in any neuroscientific study. And you see the Brodmann and the Talira atlas. Cerebellum is not there. If you read the story, they wanted to optimize the imaging space. Okay, it's cortex is there. Who needs cerebellum kind of thing? And like a lot of cerebellum studies are kind of a little bit lagged to follow up because you need a larger field of view. And before that, the subcortical areas, which is a crucial thing to work the cortex with subcortical cortical interactions, those atlases came up even way later because the imaging advances. Also, the high resolution imaging was not there. So if you want to check the history of atlases, it's kind of giving a nice overview. I put the barcode over there. It's a good reading material. Talking about brain-body interactions, let's say that's the neck. Above the neck is brain. What we do is neuroscience. We're interested in brain. Below the line there is the body, if you will. Then we have to look at the junction, how brain is connected to the body. That may give us the idea of what we can use as a peripheral measure and how we can interpret. If we just open up the neck and look at it, there are a lot of veins going there, which is the main blood supply of the brain. That could help us to track the pulse wave, perfusion. There's like a lot of sequences that tag spins there and then follow how it goes in the brain and tries to help us the global function and how the body helps how the body interacts with the brain. And that's not the one system. If you go further, it's a little bit laggy. Oops, here, I guess. Yeah, so there is another connection, which is the liquid. The liquid is fluid and produced in the spine. It's called cerebrospinal fluid. It's like circulating around the brain, and there are like certain areas, ventricles and gaps in the brain that this liquid circulates. And this is another connection. Although everything seems like happening in the skull, but the driving force is actually the heart and respiration primarily. So we can say this kind of brain-body interaction part. And then there is also lymphatic flow, which is nicely explained in the Niedergart's Beginner's Guide to the Lymphatic System. Brain is lacking of lymphatic veins, so it needed this pulsations through the heart and the veins inside the arteries and the respiration that drives these forces and then circulates around the brain. And there's a coupling with the other systems too. There's a lot more. I mean, if you look at the neck, we will see the nerves. There's two distinctions: central nervous system and peripheral nervous system. And the distinction starts right after the spine. So where the brain is connected to the external body. And then if you look at in detail, as all connections, cables, and sensors going inside the organs that is bidirectional, not only one direction. Brain can increase the speed of heart through the vagal nerve or like gastrointestinal motion, or it senses at certain pressure increase and then sends back feedback to brain through the sympathetic and parasympathetic. So this is a complex system to study with, and any isolated thing will probably be related to the rest. But yeah, we will talk about in detail. So how we measure those signals? I mean, we're just putting tons of sensors and cables around the body and trying to understand what is happening together with the brain activity. And before diving into all those signals, I mean, it could be peripheral blood pressure or like finger blood volume measures, pulsations, or like muscle tone, or like even surface pulsations that is placed on the vein can be used as a peripheral measure. We actually don't have to image all the body. We can just have a proxy measures that to try to relate with the brain. But before diving into that, I just want to give a nice anecdote and the history. This guy, I mean, his name is Angelo Mosso. It's an Italian physiologist. His work is basically trying to measure the cerebral blood flow by weight. We have basically a scale. A subject lays on the scale that is fine-tuned. There's a weight there, and this is the scale that is exhibited in the Museum of Turin. So you basically lie on this, and then he asks you a question, and he's expecting that your brain gets heavier. That was his idea. And he actually measured some pulsations without any electricity, just needle rotating on the paper without any helium or expensive MRI devices. That was kind of fascinating. But soon after he noticed that just one needle is not enough because the subject breathes, the balance is broken the other way. The subject has heart pulsations. So he just wanted to isolate the effect of the question, math question, which was the challenge right there presented, what, 150 years ago? So it's probably the challenge now is continuing. Interestingly, his work is written only in Italian till 2014. In 2014, he went viral because he is translated in English, and then a lab in Germany, I believe, replicated his work. Is this really real that your brain gets heavy when you when you ask questions or get stimulated? It turns out to be yes. I mean, they did it a little bit modern system. It's basically a flat system with some electronic scale here. And then there's like, this is the mirror. They flash some visual and then play some audio. And if you do the both, there is a weight increase on the scale related with the task. Very minimal. I don't know what it is. It's 0.5 millinewton, probably 5 milligrams. It's a global increase. That's very interesting. And recently, Marcus Raihal, which is known as the father of default mode network, wrote a book, which I put the link here, about his life story, how he tries to measure the brain function by just limited, electric scale things. And yeah, he also has a lot of devices with the peripherals, like a wearable fish tank that measures the oxygen or stuff like that. But it is fascinating that there is he be able to measure the time series that we measure with the expensive devices today. So what is up to date? I mean, it's been a lot of years after that. Things has changed. We have sensors and everything, and things is a little bit still entangled. There are a lot of factors playing a role. What is happening in the brain together? If you look at the main driven thing, for example, EEG oscillations, that's a known thing. If you study EEG, if you close your eyes, the amplitude of alpha waves changes. And that known to drive certain patterns in the brain. Moreover, like recent vigilance studies, wakefulness, drowsiness, they also known to drive similar patterns. Not only that, respiration variability, the volume, the depth, and the finger signal. You're measuring the blood volume changes in the finger. That has to have a distinct feature, which is resembling the pattern right in the middle. And there's like heart rate changes and also CSF recently, 2019 in this study. In that study, they just put the imaging slices right at the fourth ventricle, which makes it sensitive to the inflow. They didn't do any specific sequencing. It's a known fact that the edge slices are sensitive to inflow, and they measured the CSF mechanism I explained at the beginning, right there. And then that signal has a big correlates with the gray matter signal. So it seems like these systems are talking together. And the other study was by et al. 2019. It's looking at the before correlating with the fMRI. She was looking at the cross correlation of those signals, tried to get some causal inferences, like what happens first and what comes next. I think that's kind of a technique that we all need to think about it, because we all want to do the relation. Okay, you increase your breathing rate, now this increases, then maybe the EEG waves, then the PPG, which is the finger signal. So that might be a technique to start to disentangle. And looks like fMRI is the latest, put together with the PPG, but heart rate and the respiration is kind of driving. This is a study from Rasmus 20 years ago. Actually, it's been a while. This is cited a lot, the respiration changes. It is measured with the bellow shown on the left up. And you can drive two regression thing over there. It's just the bellow itself, how deep you breathe and stuff, but also the envelope of it, the amplitude of it. And actually, the blue line, if you look at it, the frequency domain looks very like the fMRI hemodynamic frequency domain. It's below 0.1 hertz. And he correlated that, which is called respiration volume. And on the left side, you see the group results with the concordance of all subjects. It's pretty much everywhere. More around the big veins, like back of the head, draining venous, but pretty much all the gray matter. And you see in the second image, you see the delays of it. So the respiration time series is delayed to match with the fMRI time series. You see the locations where it is the most affected. And I took the sentence at the end of his paper. I think it is still valid today, including these respiration fluctuations, regressor in the analysis, however, should be done with caution, because their subjects basically breathing pattern is like to vary with the task. Because if you show a cognitive task or like a on and off task, the typical tendency is, okay, the task is starting. Oh, you hold your breath. It's kind of a little bit time synchronized. So yes, if you remove the signal, you may approach a step closer to the neuronal signal, but you have the risk if the signal is task correlated. And that's what we need to check still today before removing those signals. This is also another study in our lab. There's a fourth ventricle coupling. I was also thinking that this is a peripheral measure. As I said, the fourth ventricle signal pulsations are driven primarily with the peripheral measures like heart rate and respiration. And this global changes, if you just look at this tiny red area right there, is showing a lot of information with a spectrum that is distinct for 0.05 hertz. And if you see in the middle, it gets the power gets higher because the subject is in a sleep state. And then the pulsations are increasing the amplitude. And Javier looked at it. Okay, let's compare sleep versus awake, just looking at that tiny dot, it's fourth ventricle. And he found that the certain cortical areas has some coupling with that time series. And that coupling changes sleep versus awake. And that is kind of a CSF, and you may say gray matter, but it might be a vascular component of a gray matter coupling. Right there, looks like not necessarily a neuronal activity, but maybe a systemic physiological changes. Um, this this is a study. This me and Stephanie was working on it, and this is the idea is okay. Let's not look only in the bold signal and let's tweak it a little bit so it is like sensitive to the other metrics, like CSF signal more than the bold signal. The way you do that, you just change the time of acquisition and you get different echoes, and then you have different contrast given in a TR. So one can show a little bit more CSF, the other can show a little less CSF, but a little bit more bold or blood volume. So that's the idea. If we do this with, for example, breath holding task, which we know that it drives the CSF pulsations, but by changing the contrasts, we start to see different spatial signatures. So, for example, the red one is the second interval time, which is more sensitive to blood volume, kind of a vaso sequence. You see a lot of signature around the veins, and it has high pulsation power if you look at the frequency domain. And if you look at the green, which is in the regular bold acquisition range, it's kind of scattered all around the brain with the more gray matter overlap. And the blue one, which is the early interval and early echo, is also looks like something vascular, but a little bit off, which indicates, although this is the low resolution and hard to understand, indicates might be a CSF around the veins. So this needs to be studied with the maybe high resolution to see that if it is really focused on the CSF around the veins, or is it contamination or the separation is not there? But this might be a way to disentangle right at the measurement before using peripheral measures. The other technique I recently started to use is the tiny pressure pad, which is very sensitive to changes, extremely to motion. It needs to be placed very close to the carotid artery, which is if you put your finger on your neck, you will feel it already, and then you get this kind of a signal. This looks a touch different than the signal we measure from the finger. The one we measured in the finger has different features. The one we measured in the neck has different features. And most importantly, they're a bit delayed. They're not in sync. And we can also look at the breathing because breathing can change these things too. So if you look at zoom in closer, like there's two signals, one from the neck, one from the finger, you will see the delays of, I don't know, 30, 40 milliseconds, which is the blood arrival time, which arrives on the neck earlier than the finger. And this I will talk more about on next Tuesday, more under the hood technique, how we do the peak detection, how we separate the signal. Is a part of apnea. Basically, you need to detect when the peak happens, in both peripherals, and then you look at how different they are. So if you look how different they are, basically peak-to-peak delays in the two things, you get a signal like this, and the signal says how the delay changes every pulse. So it's basically pulse transit time. It is variable because veins can dilate, heart can pump faster, and so on and so forth. You see the histogram at the bottom, so it's pretty scattered. And I put the expected time of delays. The top is the ECG, with the electrical measure of the heart. And there are like certain delays. You can even measure the pulse from the toe, which will be the latest. So why we do that? Because we want to know this vascular changes in the periphery and try to relate with the brain because it may comply. I mean, brain is attached to the body. So take it as a pinch of salt. This is like a very early primary preliminary results. We see that positive correlation in the frontal brain and negative correlation at the back. you can make an interpretation story out of it. Yeah, arteries dominance in the front, maybe is the increase the time of arrival, the slower pulse wave arrival, and longer arterial expansion and then increase in bolus. Or it's but basically maybe just delay. So it is like positively correlated front, negatively just because it's the phase delay, which needs to be investigated. What also needs to be investigated is all with the multi-echo that then explained a couple of weeks ago. I put the link over there to Tadana to see that if these signatures will survive as a bolt signal, or they are maybe the CSF modulations, which is kind of coupled with the peripheral vascular pace. These are important questions because we need to understand this is during the rest. There's subject is doing no task. So the measure the signal we are measuring, we want to know if it is just physiology basis or is it just background neuronal thing. But not to remove and just to leave it the neurons, but also to understand the brain mechanism was also a good question too. So this might be a way to study brain-body interaction too. Next, I will talk about pulse oximeter. I mean, it is insanely rich signal that is highly studied, but less studied with fMRI. I mean, it has a lot of features. Time to rise, systolic peak, the second peak is diastolic. If second peak is lower, there's some vascular differences. It could be broadened or short, and it's measured with the light. There's a finger showing over there. The one we have is the reflective one, so the receiver is there. It changes the feature a little bit, but so there's the base, venous blood, so there's pulse style component on it. And one can extract tons of information out of there. So the location of the pulse changes the second peak, because if it's further, the reflection is kind of attenuated. If it's close to heart, you see the second peak, which is the diastolic phase of the heart. But that's why when I place the pulse on the neck, I see the second peak too. Though most of the people will study just the delays and the peak and the amplitude, but if you isolate the individual peaks, you get subtle changes of rise time, fall time, and expansion, which are actually related with the blood pressure, which I will dive into in a bit. So yeah, blood pressure is a clinically very valid measure that is typically measured with the cuff spigometer. That takes about a minute or so. You need to inflate it and kind of pinch the artery, make the coupling, and hear the sound through the stethoscope, and then understand when it is coupled to understand the systole and diastole. Or invasively with the IV that is coupled too, which is not an option. And yeah, every heartbeat could be different, and it takes time to arrive to other body parts. And actually, blood pressure is all variable around the body. So when we say blood pressure, it's the average of a cycle. And how we measure the MRI is a different technique, which I mentioned a little bit in the PPG. So there's a device that measures the pressure cuff around the finger that is very sensitive to the second peak, which is the blood pressure estimation is happening. It's called reflectance of it. And it is quite decent from the finger. It's not completely attenuated. And when the pressure changes, that peak goes high and low. It's basically internal derivative algorithm. And then in this study, they just show that it doesn't show significant difference to regular blood pressure measures. That this is me trying that if device is really working. There's a device that I try to increase my chest pressure and see how it changes the PPG signal. Pulse looks like the second peak is there if I increase my chest pressure. It is an interesting phenomena. When you increase the chest pressure, blood pressure goes down. No one believed that at the first, But after digging the literature and trying to understand what's happening in the body, when you see the image at the bottom, the vein vena cava is piercing through the diaphragm to go to the heart. So when you squeeze your diaphragm, you're actually restricting return blood, which creates a vasodilation because body thinks about blood is not coming to the heart, so you need to dilate it. And the pressure goes down. It's a transient thing, and it goes up again. That might be the reason why meditation, breathing kind of things relax you. But yeah, it's a good news that we can modulate it, but it's a compound modulation because respiration has other modulative things on the brain too. It's not super isolated. So the blood pressure has multiple components, but the way we measure is from the finger, which means like it is dependent on the heart rate. Some subjects are 50 BPM, and your sampling is low because heart is beating slow, because it's estimated every heartbeat. So the low frequency component is the vascular tone, which has a huge effect on the fMRI side. But the sympathetic part and the kidney part is very high pressure, and it's not accessible to non-invasive measures. And even if we try a little bit with the high heart rate subjects, it's a very noisy measure that doesn't show much in the brain. Moreover, we can look at how increased blood pressure or increased pulse wave correlated with the brain. So you see the delays versus correlation value. You can tell that the finger pulse, which is a blood volume proxy, goes higher with the correlation and with the higher, longer amount of delay. On the other side, blood pressure is kind of short-lived pulsations happening around a few seconds. And we looked at how it is connected to the brain, the delay structure. The top is the finger PPG signal. The bottom is which is the mainly blue, happening around the zero, short-lived, although it has some signature. It looks like default mode network that is a little bit pressure driven. Not, but we can have external modulation. I mean, all the measures I explained you till now is like the passive listening, what's happening but we can interfere in the system. And there are ways to do that, which I'm trying to utilize. The first one, I'll talk about light stimulation, which is kind of new in Gene Chan's group. I put the barcode over there. We have the system. You basically shine a light inside the nose and hits at the brain and creates a vascular response because a lot of light sensitive proteins that creates this response. And then they measured this CSF, which is very low but steady fourth ventricle pulsation after the light is shined. So you can create actually at the fourth ventricle a response by just shining the light. And the other one is the pressure cuff, which you just restrict the blood flow to a limb. And then that will have change in blood distribution in the body because you just block it there and then maybe it triggers vasodilation because blood needs to go other way more freely. So we looked at too. And the last thing is the vagal nerve stimulator. It's an interesting one. If you look at the anatomy, the part of the ear has a branch of the vagal nerve. And you need to place it such that the stimulation goes into the vagal nerve, and then you hope that it changes the sympathetic and parasympathetic balance. But most of the studies are doing like a therapy kind of thing. So you get a stimulation for, like, say, half an hour, then you go in the scanner kind of thing. But we have a system that goes in the scanner. We just don't know at this moment. It's just single stimulation will cause any significant sympathetic parasympathetic balance change. And the other thing is there are a lot of things may change in the body just because you stimulate. You may not hit the vagal nerve branch. So there should be a null area, which is in the sham. It's kind of closed area that you stimulate there just for the basic baseline. So those are the ones we're trying to utilize in the lab. We already tried the one with the leg pressure cuff setup. And subject went in, then there's like, this is a kind of variable cuff. We inflated the one around the, there are two compartments that goes around the calf muscle that obstructs with 100 mm mercury. So that's above the diastolic pressure. So the venous blood is basically kind of isolated in the leg, but a small portion. So there's a compressor here, and there's a control unit, and then subject is going under a regular fMRI scan. And we basically inflate the cuff such that in different durations, like 30 seconds, 100 seconds, 50 seconds, and we see a lot of scattered and isolated response along the brain, primarily in the central ventricles, positive and negative neighboring the walls of the ventricle, which indicates kind of a CSF expansion that we need to check. And also a lot of in the gyri and sulci. If you close look here, like there's a black dot, which is probably wane, and there's like a negative response there, and there's another wane here, there's a negative response there. So we're still investigating what that happens in the leg, then causes huge fMRI response changes that is not sensory. Looks like location-wise, it is more like compliance changes in the brain due to the blood restriction. And here we see the significant voxels around across the slices. It is usually the CSF around and central. And if you go further down, there's a bit on the fourth ventricle too. And interestingly, eyes too. That was really shocking. I was not expecting that eye time series will follow the cuff pressure. I mean, there's no physiological explanation that eye will move with the cuff. So it is probably a eye tension. That, I mean, the subject was fixating during the scan, so eyes are less likely to move with the cuff pressure. But it may be well with the MR noise that is caused by the inflation that we need to check that too. We further try to understand that if this is so dominant that it will override the visual checkerboard. So here I show a signal that subject went through visual task, which is flickering checkerboard. And here I have zoomed in. It actually, this cuff signal is dominated over the visual signal. So it probably changes the blood flow at the visual cortex at the back of the head more than the visual task does. So that kind of emphasized the importance of peripheral changes, maybe more stronger even in some cases than the neuronal task we're trying to measure. I'm getting close to the end. I would like to stress the motion. I mean, yes, the measures are for fMRI, but the motion in the brain is not well studied. I'm talking about small motions like wane expansions, CSF compartment compression, partially related with the glymphatic system, because glymphatic system is the water exchange in between these areas, which cause this the motion changes. And there's a lot of water around the brain, which helps brain to move actually. And if you look at the, brain surgeries, brain is pulsating. If you look at cardiac gated motion camera, skull is moving too, but brain is moving in the skull. That's the motion type I'm talking. And even surgeons having a hard time to placing an electrode because brain is kind of wobbly with every heartbeat. It is also a slow wave too. So that might be a source of information that we were looking for, which I'm kind of utilizing a code that works on volume by volume, how brain shape changes. It might be an informative way to look at it, because that's usually not used in the fMRI. What we use is rigid body that just doesn't take account of this local expansions and things. So if you look at here, for example, fixed and moving, what you're trying to do is you try to put the moving part warped to look like a fixed one. So you basically expand certain areas to look like all the gyri or sulcis are matching, which is kind of a way to find what is the motion difference in between two volumes. And it gives completely different 4D time series, because if you do that, now some time series are more, could be, which is still undergoing, more task related than the original time series, because those are the motion related. And if you do this morphing, then you amplify the motion sensitivity that might be help us to relate with the all peripheral measures with the task. I have a very preliminary result with the two different tasks, breathe in and hold and out and hold. In and hold is the red one with the same subject, which causes this motion changes, significant areas, which is in the central brain area, around the ventricles, and the green one is the inhale and hold. So it looks like, depending on the task you're doing in your respiration, it's changing how brain morphs itself. With that, I will close. That's all I have for today. So I think the take-home message will be, yes, measuring peripheral physiology provides essential context to interpret a little bit better what we are seeing in the brain. That's for sure. And understanding the origins of MRI signal is very important, because what we can see could be an artifact or like indirect measure. So that's very crucial with the physiological interactions. I think it's key to understand the brain function. And multimodal imaging, like adding some echo time differences to increase the sensitivity of one thing, may help us to disentangle. I don't want to say it's like removing physiology or like getting better neural signal. I will say physiology aware fMRI. That's the term I want to add at the end of this talk, being aware of what's happening. And the task design becomes increasingly more important, because, as I said, breathing may be task at the time lag with the task. So we may need to do a better task design. We may even do two simultaneous task designs, which is like more vascular, independent, more task related. And I think I'm confident to say brain cannot be fully understood without understanding the body. It's a part of the system. So with that, I'd like to finish my talk. Happy to answer any questions you might have. Thanks.
Question about task design. So if you're concerned, as you're sort of breaking up the study, that the effect that you're seeing in the bowl or your fMRI signal isn't the neural event you're interested in, but differences in breathing behavior, let's say, between condition A and condition B. How do we mitigate that? Are you suggesting that we record all event physiology as best we can and try to regress it out later? Or should we be exploring approaches where we have subjects, like in a perception task or in a cognitive task, asking them to maintain, breath during a time period of interest, so you can sort of remove potential differences in that behavior?
Yeah, I think it depends on the what you study and how you study, because if you know the frequency of breathing, and if you can let your subject do breathing tasks together with the cognitive task, you may control the timings separate apart, that it's like frequency separable or time separable. You can design that, or you can just record that and be sure that as a QA metric, hey, 20% of my subjects are time locked with the respiration. There's like huge activity area, so I cannot disentangle that. That might be the way. It just depends on how engaging, how triggering your task is with the breathing. I think that's an issue with the more cognitive load tasks because you focus on and then you hold the breath. But if it's just a flickering checkerboard, the effect might be less. That I don't know. Yeah. But yeah, breathing and the finger pulse is available in all MR scanners we have. So will be good to acquire. Any questions online? Online people are not there anymore. All right. I think if no questions, that's it for today. See you on Tuesday.